Molecular evolution. The scientific theory of evolution by natural selection was conceived independently by Charles Darwin and Alfred Russel Wallace in the mid 19th century and was set out in detail in Darwin's book on the origin of species. Biochemical evolution was the first step in the development of life on Earth. The formation of complex organic molecules from simpler inorganic molecules through chemical reactions in the oceans during the early history of the Earth. 5 lines of evidence that support evolution: the fossil record, biogeography, comparative anatomy, comparative embryology, and molecular biology. Evidence that support the theory of evolution by natural selection: biochemistry is the study of basic chemistry and processes that occur in cells. Biogeography, the study of living things around the globe, helps identify Darwin's theory of biological evolution. Micromolecules of life: pyrimidines, purines, fatty acids, macromolecules or monosaccharides, amino acids, nucleotide. Once again, pyrimidines, purines, fatty acids, monosaccharides, amino acids, nucleotides. Molecular evolution is the area of evolutionary biology that studies evolutionary change at a level of the DNA sequence. It includes the study of rates of sequence change, relative importance of molecule, adaptive and neutral change, and changes in genome structure. The process of change in the sequence composition of cellular molecules such as DNA, RNA, and proteins across generations. The field of molecular evolution uses principles of evolutionary biology and population genetics to explain patterns in these changes. The technique used to study molecular evolution are based on the comparison of nucleotide and amino acid sequence among species. Data from embryos can be used to help establish evolutionary relationships. 1962, Linnaeus Pauling and Emily Skrkencil proposed using the number of differences between homologous protein sequences to establish the time since divergence, an idea Skrkencil had received around 1960 or 1961. Genetic divergence. Divergence pertains to an evolutionary process wherein a population of an inbreeding species diverges into two or more descendant species that have become more and more dissimilar in terms of forms and structure. Eventually, they become different species to their ancestors. Divergent evolution leads to speciation and works on the basis that there is variation within the gene pool of a population. One possible cause of genetic divergence is the founder effect, which is when a few individuals become isolated from their original population. Those individuals might overrepresent a certain genetic pattern, which means that certain biological characteristics are overrepresented. And molecular phylogeny. Molecular phylogeny is the relatively new scientific discipline that involves the comparative analysis of the nucleotide sequences of genes and amino acid sequences and structural features of proteins from which evolutionary histories and relationships, and in some cases also functions, can be inferred. And molecular similarities provide evidence for the shared ancestry of life. DNA sequence comparisons can show how different species are related. Fossils provide evidence of long-term evolutionary changes documenting the past existence of that species that are now extinct. Molecular systematics is the use of molecules to determine classification systems and relationships. For hundreds of years, botanists use morphology or ovular appearance to identify the class and classify plants. Molecular phylogenetics is the branch of phylogeny. That analyzes genetic hereditary molecular differences predominantly in DNA sequences to gain information on an organism's evolutionary relationships throughout various branches in the tree of life. So molecular biology often helps in determining genetic relationships between different organisms. In particular, the sequence of a small subunit ribosomal RNA is widely used in molecular phylogeny. Some of the best evidence of evolution comes from examining the molecules and DNA found in all living things. A chicken and a gorilla will have more differences between their DNA and amino acid sequence than a gorilla and an orangutan. A molecular clock. A molecular clock is a figurative term for a technique that uses the mutation rate of biomolecules to reduce the time in prehistory when two or more life forms diverged. The biomolecular data used for such calculations are usually nucleotide sequences for DNA, RNA, or amino acid sequences for proteins. A molecular clock does tell time. on an epoch scale, a tool used to calculate the timing of evolutionary events by Blair Hedges. Evolutionary biologists can use this information to deduce how species evolve and to fix the date when two species diverged on the evolutionary timeline. Over the course of millions of years, mutations may build up in any given stretch of DNA at a reliable rate. For example, the gene that codes for a protein alpha globin, a component of hemoglobin, experiences base changes at a rate of something. If this rate is reliable, the gene could be used as a molecular clock. And measuring the age of species with a molecular clock technique requires just two simple things: an estimate of the number of genetic mutations between a species and its closest relative, and the average genetic mutation rate, how many mutations show up in a population in a specific time frame. The molecular clock hypothesis assumes a constant rate of change in sequence over evolutionary time, has often been used in the interpretation of trees. Then molecular clock, much more hierarchic than previously thought and practically useless to keep accurate evolutionary time, and researchers conclude they attribute this to the vagaries of natural selection, which may at times constrain specific genetic mutations in certain lineages. The erratic manners arises questions of their use and even the entire theory of evolution. Molecular clock model determining relatedness between species are it assumes that all changes in amino acid sequence are random and are not affected by natural selection, which is not true. The evolutionary process wherein a population of species diverge into two or more descendant species, resulting in one similar or related species become more and more dissimilar. Then, in the light of uncertainty in fossil dating and the rates of evolutionary change, the molecular clock cannot provide high levels of precision, but when used correctly, it can still prove accurate. The lack of precision reasonably reflects our uncertainty about the past. Molecular systematics, the use of molecular genetics to study the evolution of relationships among individuals and species. The goal of systematic studies is to provide insight into the history of groups of organisms and evolutionary processes that create diversity among species. Then phylogenetics, the science of estimating and analyzing evolutionary relationships. Phylogenetic relationships among microorganisms are especially difficult to discern. Molecular biology often helps in determining genetic relationship between different organisms. Then molecular chronometer, a phylogenetic marker, a highly conserved protein, ubiquitin, or nucleic acid and rRNA whose rate of mutation is constant and which can therefore be used to construct phylogenetic trees. Then genetic equidistance. Genetic equidistance. The genetic equidistance phenomenon shows complex taxa to be all approximately equidistant to a less complex species in amino acid percentage identity. A genetic equidistance phenomenon shows complex taxa to be all approximately equidistant to a less complex species in amino acid percentage identity. The overlooked mystery was interpreted by the maximum genetic diversity hypothesis. Molecular dye, term coined by Gabriel Dover in 1982 to describe evolutionary processes that change the genetic composition of a population through DNA turnover mechanisms. The evolution of living organisms is the consequence of two processes. First, evolution depends on the genetic variability generated by mutations, which continuously arise within populations. Second, it also relies on changes in the gene frequency of alleles within populations over time. Then, meiotic drive occurs when processes during meiosis or later stages of gametogenesis skew that ratio such that one allele, the driving allele, is overrepresented in the gametes. T is sometimes also called transmission ratio distortion or segregation distortion. Then neutral theory of molecular evolution. The neutral theory of molecular evolution intends that at the molecular level, most evolutionary changes and polymorphisms within species are not caused by natural selection, but by random genetic drift. The theory was first put forward by Kimura in 1968. Because of natural selection, advantageous mutations have a higher probability of fixation than neutral mutations, and deleterious mutations have a lower probability of fixation. Neutral theory of molecular evolution was first proposed by Moto Kimura in 1968 and independently by Jack King and Thomas Jukes in 1969. The neutral theory of molecular evolution contends that most evolutionary change at the molecular level is driven by genetic drift rather than natural selection. The neutral theory can be contrasted with the idea that almost all molecular evolution has been driven by natural selection. Then the neutral theory of molecular evolution holds that most evolutionary changes occur at the molecular level and most of the variation within and between species are due to random genetic drift or mutant alleles. Random genetic drift and mutant alleles that are selectively neutral. One of the original shortcomings was that the neutral theory could not explain the varying patterns of genomic evolution observed among species with different population sizes. In small populations, however, purifying selection is less effective and allows slightly deleterious mutations to behave neutrally. Eventually, it boils down to whether the neutral theory should officially be pronounced utterly dead or if it still has its space as formulated by Kreitman more than 2 decades ago. Kreitman 1996. It is my thesis that the neutral theory and non-coding variation will continue to play a leading role in the test. And orthogenesis. Orthogenesis is the assumption that evolutionary changes or variations are produced in straight lines. The observed tendency of a part or organ to change progressively in size is called orthogenesis or evolution in a straight line. The theory of orthogenesis was proposed by Emir in 1897. Then orthogenesis, vitalism, Lamarckism, then idealism, holistic evolution, structuralism, mutationism, modern synthesis, and Darwinian evolution. These are related. Orthogenesis, also called the straight line evolution theory, that successive members of an evolutionary series become increasingly modified in a single undeviating direction. A variety of theories have attempted to explain orthogenesis, but with only partial success. A variety of theories have attempted to explain orthogenesis, but only with partial success. An underlying view in orthogenesis by some paleontologists is that variation in nature is directed towards fixed goals and that species evolve in a predetermined direction irrespective of selection. And collapse of this hypothesis. The orthogenesis hypothesis began to collapse when it became clear that it could not explain the patterns found by paleontologists in the fossil record, which was non-linear with many complications. The hypothesis was generally abandoned when no mechanism could be found that could account for the process, and the theory of evolution by natural selection became the prevailing theory of evolution. And the modern evolutionary synthesis in which the genetic mechanism of evolution were discovered refuted the hypothesis for good. As more was understood about these mechanisms, it becomes obvious that there was no possible naturalistic way in which the newly discovered mechanism of heredity could be far-sighted or have a memory of past trends. Variations depend on the constitution of the varying organism. A modification, whether it be a large or small, is a modification of an already definite and limited structure. A continuous environment, both from the point of view of production of variation and selection of variation, would appear necessarily to result in a series with the appearance of orthogenesis. The history of organic world displays many successful series, and this, as they have survived, most inevitably display orthogenesis to some extent. But it also displays some failure, which indeed may be regarded as showing that imitation of variations has been such that the organism have lost the possibility of successful response to new environment. A few hung on to the orthogenesis hypothesis as late as 1950s by claiming that processes of macroevolution, the long-term trends in evolution, were distant from the processes of microevolution, genetic variation, and natural selection. Microevolution in the sense genetic variation and natural selection, which were by then well understood, and it was known they could not behave in a orthogenetic manner. They Sheridan, a Jesuit paleontologist. The Phenomenon of Man, a book influential among non-scientists that was published 4 years after his death in 1959, argued for evolution aiming for the omega point while putting man at the center of the universe and accounting for original sin. Then the term Chardin used for this was directed addictivity. This form of orthogenesis for now also been abundant as more about evolutionary processes has been discovered. The refutation of orthogenesis had some ramifications in the field of philosophy, as it refuted the idea of theology as first postulated by Aristotle and accepted by Immanuel Kant, who had greatly influenced many scientists. Primates evolution, forward facing eyes, stereoscopic vision, bony orbital eye rings, then reduced snout and olfactory apparatus. Once again, forward facing eyes, stereoscopic vision, then bony orbital eye rings, reduced snout and olfactory apparatus, then middle ear enclosed by an outgrowth of the petrosal bone, then large and complex brains compared to other animals, independently moving digits with sensitive ends, opposable thumbs, and except for humans, opposable large toes. Nails on one digit at least, a clavicle or collarbone, two separate bones in the lower forelimbs, ulna, radius, and the upper hind limbs, tibia, fibula. And primates are organized into two groups, haplorhines and strepsirrhines. Haplorhines and strepsirrhines. Haplorhines and strepsirrhines. The word haplorhine translates to dry nose and the word strepsirrhine translates to wet nose. The haplorhines or dry-nosed primates include monkeys, apes, humans, and tarsiers. Primates are organized into two groups, haplorhines and strepsirrhines. The word haplorhine translates to dry nose and the word strepsirrhine translates to wet nose. The haplorhines or dry-nosed primates include monkeys, apes, humans, and tarsiers. The wet nose of animals called a rhinarium is a bare and wet because it can greatly improve its olfactory system or sense of smell. This allows water to evaporate from their nose and cool them off. When the tiny sand particles in the air are dissolved in water, they are more likely to set off the smell detectors in animals' nose. Humans are mammals too, but we belong to the primate group that doesn't have wet noses. Rhinarium or New Latin, belonging to the nose, is the furless skin surface surrounding the external openings of the nostrils in many mammals. Informally, it may be called the truffle wet snout or wet nose because its surface is moist in some species, for example, healthy dogs and cats. The lower primates or strepsirrhines, suborder Strepsirini, include lemurs, bush babies, lorises, and higher primates or haplorhines, suborder Haplorhini, include the tarsiers, old and new world monkeys, apes, and humans. Strepsirrhines have moist noses, haplorhines have simple dry noses. The lower primates or strepsirrhines, suborder Strepsirini, include lemurs, bush babies, lorises, and higher primates or haplorhines, suborder Haplorhini, include the tarsiers, old and new world monkeys, apes, and humans. Strepsirrhines have longer snouts, smaller brains, and more highly developed sense of smell than haplorhines. Haplorhines have shorter faces, larger brains, and more highly developed sense of vision than strepsirrhines. Their eyes face more forward than the eyes of strepsirrhines. The different lines of primates show gradual changes in the hands and feet, the evolution of sensitive pads for gripping on the underside of fingers and toes, and increased mobility of the digits for grasping objects. These changes culminated in the development of human hand and a thumb that can rotate to oppose the fingers. Adam's Eve, both mitochondrial DNA and Y chromosome have been used extensively by molecular paleoanthropologists in attempt to construct human lineages. Both are inherited in haploid manner. mtDNA through the female and Y through the male. So mitochondrial DNA is separate from nuclear DNA that is contained within the mitochondria, intracellular organelle that provide energy to the cell. Its inheritance is analogous to the Y chromosome, with the DNA inherited unchanged apart from mutations from the mother. In sexual reproduction, during the course of fertilization event, only nuclear DNA is transferred to the egg cell, while rest all other things are destroyed. And this is the reason which proves that mitochondrial DNA inherited from mother only, maternal inheritance. The mitochondrial mtDNA contains genes involved in the process of oxidative phosphorylation, the production and storage of energy. This is evidence that mitochondria once existed as a free-living bacteria which were taken up by primitive ancestors of eukaryotic cells. Mitochondria in mammalian sperms are usually destroyed by egg after fertilization. In 1999, it was reported that paternal sperm mitochondria containing mtDNA was marked with ubiquitin to select them for later destruction inside the embryo. So there must be some ubiquitination happening in case of this mitochondrial sperm mtDNA. Mitochondrial DNA is a naked double-stranded circular extra chromosomal genetic element continuously exposed to the matrix that contains great amount of reactive oxygen species and free radicals. Mitochondrial Eve is a female biological ancestor of humans, aptly named the mother of all humans. It might seem very unusual or even impossible, but the DNA inside the mitochondria explains everything. There is one DNA that a human child inherits from the mother. So in most metazoans, mtDNA shows an elevated mutation rate compared with the nuclear DNA, likely due to the less efficient DNA repair, a more mutagenic local environment, putatively caused by oxidative radicals, and an increased number of replications per cell division. DNA evidence has revealed that the oldest common known male ancestor is 340,000 years old, more than twice as old as previous estimates. New scientists report that the sample came from a recently deceased man named Albert Berry. The most important advantages of using mtDNA is its intrinsic ability to resist degradation and its high copy number inside the cell as compared to nuclear DNA. High copy number. Each cell contains around 1000 mitochondria and there are 2 to 10 copies of mtDNA per mitochondria. So 10 into 1000. Now a term of researchers led by Cosimo Post from the University of Tübingen in Germany analyzed the DNA of an ancient skull belonging to a female individual called, I'm not sure about the pronunciation, Slatikin, and found that she lived around 47,000 to 43,000 years ago, possibly the oldest genome identified to date. In fact, there is a whole lot more to maleness and femaleness than X or Y chromosomes. About 1 in 20,000 men has no Y chromosome, instead having 2X. This means that in the United States there are about 7,500 men without a Y chromosome. Humans first evolved in Africa and much of human evolution occurred in that continent. The fossils of early humans who lived between 6 and 2 million years ago come entirely from Africa. Most scientists currently recognize some 15 to 20 different species of early humans. The earliest record of Homo is the 2.8 million years old specimen LD 351 from Ethiopia, and the earliest named species are Homo habilis and Homo rudolfensis, which evolved by 2.3 million years ago. Mitochondrial Eve is a term for genetics and human evolution. It refers to a woman whose mitochondrial DNA is a parent of all living humans. To put this more exactly, she is the female most recent common ancestor to all humans alive at present. Mitochondrial Eve, mother of all humans, lived 200,000 years ago. Summary: the most robust statistical examination to date of our species genetic links to mitochondrial Eve, the maternal ancestor of all living humans, confirms that she lived about 200,000 years ago. In human genetics, the mitochondrial leaf, also MTE, MTMC, MRCA, is the matrilineal most recent common ancestor. Matrilineal most recent common ancestor of all living humans. As the identity of both matrilineal and patrilineal MRCAs is dependent on geological history, pedigree collapse, they did not have lived at the same time. Evolution of communication. One thing we can see in the remains of early humans, however, is the development of the mouth, throat, and tongue. By about 100,000 years ago, humans had evolved the ability to create complex sounds. Before that, evolutionary biologists can only guess whether or not early humans communicated using more basic sounds. The gestural theory states that human language developed from gestures that were used for simple communication. Two types of evidence support this theory. Gestural language and vocal language depend on similar neutral systems. The regions on the cortex that are responsible for mouth and hand movements border each other. Oldest known form of communication were cave paintings. After them came pictograms that eventually evolved into ideograms. Pictograms and ideograms. Fast forward to 3500 BC, and the first cuneiform writing was developed by Sumerians, while the Egyptians developed what is known as hieroglyphic writing. Early humans could express thoughts and feelings by means of speech or by signs or gestures. They could signal with fire and smoke, drums or whistles. As soon as the words were spoken, the gestures was made, or the smoke was blown away by the wind. They were gone and could not be recovered except by repetition. Since the dawn of time, even before we developed languages, we strive to communicate. We wanted each other. We warned each other of danger or showed each other how to hunt. After that, or around the same time, we started drawing. We have been drawing for 32,000 years. The oldest known form of communication were cave paintings. After them came pictograms that eventually evolved into ideograms. Pictograms and ideograms. Fast forward to 3500 BC, and the first cuneiform writing was developed by Sumerians, while the Egyptians developed what is known as hieroglyphic writing. The alphabet was developed around 1500 BC by the Phoenicians, and paper was only invented in Thailand in 905 AD. Some of the oldest forms of human communication include talking or making sounds, drawing or painting, dancing, acting, and using symbols. The Ponto-human language, also called Proto-sapiens, Proto-human language, Proto-sapiens, Proto-world, is the hypothetical direct genetic predecessor of all the genetic world's spoken languages, Proto-human language. It would not be ancestral to sign languages. Language allows us to share our thoughts, ideas, emotions, and intention with others. Over thousands of years, humans have developed a wide variety of systems to assign specific meanings to sounds, forming words and systems of grammar to create language. Early humans could express thoughts and feelings by means of speech or by signs or gestures. They could signal with fire and smoke, drums, or whistles. Three early methods of communication had two limitations. First, they were restricted as to the time in which communication could take place. Then, Proto Indo European languages. The Indian hypothesized mother language of all languages in the Indo European family. Proto Indo European language. This language is thought to have been spoken around 3500 BC by nomads living in what is present day Ukraine. A long popular theory of the development of the larynx, first advanced in the 1960s, held that the evolutionary shift in throat structure was that enabled modern humans and only modern humans to begin speaking. Language started 1.5 million years earlier than previously thought, as scientists say Homo erectus were first to talk. A new study that relies on brain imaging of cerebral blood flow suggests that human speech and complex tool making skills emerged together almost 2 million years ago. The earliest evidence of Homo erectus dates to around 1.9 million years ago, meaning the timing of the first language could be pushed back considerably. Professor Everett argued a primitive form of conversation could have been necessary for Homo erectus to achieve that what they did.